Hi everyone, my name is Steve Hughes. I'm the Chief Medical Officer of Avidity Biosciences. We're a California-based biotech company, and our mission is to profoundly improve patients' lives by revolutionizing the delivery of RNA therapeutics. So you will see that across our portfolio, all of our programs are in various forms of RNA therapeutics, using a novel new delivery technology called the antibody oligonucleotide conjugate. So today we're here to talk about FSHD, or Facioscapulohumeral Muscular Dystrophy. We have a program in FSHD where we just read out some data just a couple of months ago, and we've now transitioned that program into the potentially registrational phase. We've just initiated a biomarker cohort, and in just a moment, I'll tell you a little bit about the biomarkers, but the biomarker cohort is targeting an accelerated approval with FDA. And then in the first half of next year, we'll initiate a phase 3 functional endpoint cohort that will be targeting full approval globally. So FSHD is a relentlessly progressive neuromuscular wasting disease. So it's a muscular dystrophy. It is caused by aberrant expression of a gene called DUX4. The DUX4 gene is normally only expressed in the embryonic stages during early cell division and then gets switched off. Patients that have FSHD have abnormal expression of DUX4 after birth. DUX4 is a transcription factor. It regulates a number of downstream genes, several hundred in fact, and because it's a embryo-fetal transcription factor, it switches on a number of embryo-fetal genes, and the products of these genes are toxic to the muscle. It's sporadically expressed, which means that when it's abnormally expressed, it isn't abnormally expressed all of the time. It's abnormally expressed in spikes, but it then turns on all of these downstream other genes that cause a cascade of events with amplification of the signal, and that leads to a stochastic progression in these patients, often asymmetrical. What we see is often they'll get a misdiagnosis that something will be put down to growing pains or a trapped nerve or something. Whereas, the tragedy of missing the disease means that it's diagnosed late, means that genetic counseling can't be done till late, and as a whole impact, other family members can't be screened until late. So really erring on the side of caution. There are genetic tests available for all of these diseases that we're in. And so getting patients referred for genetic testing early will really help with getting the early diagnosis, and that can help with early treatment, particularly as now there will hopefully be real treatments available for these patients where none were available before. So because DUX4 regulates a number of downstream genes, we can measure the downstream gene signature of when DUX4 is switched on. Unfortunately, when DUX4 itself is expressed in very low abundance and in only about 1 in 1,000 cells when it is expressed, and so it's not really possible to measure DUX4 itself, but we can measure the downstream gene signature from DUX4, and there's a number of different panels that have been developed by different people for this. So what we showed when we disclosed our data was that by administering del-brax, or AOC 1020, which was the original name for the drug, we can basically reduce this downstream gene signature by over 50%. And we evaluated it using a number of different panels, and all of them showed a consistent greater than 50% reduction of the downstream gene signature, showing us that we're profoundly reducing DUX4. We were also able to develop in-house a circulating biomarker that's never been done before. This is actually a protein that circulates in the blood. It's only seen at very, very low levels in normal people, but in people with FSHD, it's expressed and circulates at considerably higher levels. And we were able to show that we had a greater than 25% reduction in this circulating biomarker, which we know is DUX4 regulated. And the beauty of the circulating biomarker is that it allows us to track what's happening over time with the DUX4 expression. Whereas the other biomarkers that we showed where we saw the greater than 50% reduction, those were in a muscle biopsy. there's a limit really to how many muscle biopsies you can take in these patients. For one, they're losing muscle, so you don't want to take too much. And secondly, muscle biopsies are quite painful. So this allows us to track over time exactly what's happening with DUX4. The other thing that we saw in concert with the reduction in the circulating biomarker, we also saw a reduction in creatine kinase, which is a circulating biomarker measure of muscle damage. So we saw reduction in a broad stream of DUX4 regulated genes in the muscle biopsy, measuring it using multiple different methods. We saw a reduction in the circulating biomarker, and in concert with the reduction in DUX4 expression, we also saw a reduction in creatine kinase, indicating an improvement in muscle health. So all of those things joined the dots very, very nicely from the disease biology. The other thing that we looked at using RNA-seq was what happens downstream of the DUX4 regulated genes. So we did bulk RNA-seq and we subtracted out all of the genes that we knew were DUX4 regulated. And what we were left with was a signature that was the underlying biology of the disease. What happens when you turn on DUX4 and it's downstream genes? This is what happens. And we showed that we were able to normalize that gene signature as well. So we've shown that we can ameliorate DUX4, we can affect the downstream DUX4 regulated genes, we can affect the disease biology. That turns into reductions in the circulating biomarker and also reductions in creatine kinase. So that's a very nice package, and we think this is a perfect disease for an accelerated approval. Accelerated approval is a mechanism within the United States, so regulated by FDA, to bring promising new drugs to patients more quickly than can be done with traditional clinical trials, because it allows you to get a registration and approval based upon a biomarker endpoint. The biomarker endpoint has to be reasonably likely to predict improvement in the clinical features of the disease. And the other thing that we saw in our clinical trial was that we saw trending improvements in reachable workspace, which is an endpoint that's being used quite a lot in this disease, and also in muscle strength, looking at both the upper limb and lower limb muscles in a composite and individually. Improvements in patient-reported outcomes and improvements in physician-reported outcomes. So you can see we've got a very nice package that is all consistent, showing improvement in multiple measures, both in the biomarkers, the biology, and the physical symptoms of the disease, and how the patients and the clinicians are feeling about the disease. So we think that pursuing the accelerated approval pathway using our biomarker cohort allows us to get this drug to the broader patient population as quickly as possible. We think it's a perfect disease for an accelerated approval route because the underlying cause of the disease is known. It's DUX4, have an expression of DUX4. There isn't another cause of the disease. We can measure the downstream effects of DUX4, so we know that we're ameliorating that DUX4 expression, and we've shown that that comports with improvements in, or at least trends to improvement in the clinical function in this first study. So our biomarker cohort is a larger cohort than the cohorts we've already gone to patients in. It's about 48 patients in size and allows us to more definitively show these improvements. And then our goal is to go to FDA with that data package and submit for an accelerated approval. So if you get a accelerated approval, you have an obligation to conduct what's called a confirmatory study, looking at more traditional endpoints. And that study will get underway in the first half of next year. So really, we're massively expediting this program in line with our philosophy of getting promising drugs to patients as quickly as possible. The outcome for the biomarker cohort, so that the primary outcome is the DUX4 regulated genes, both in the muscle biopsy and the circulating biomarker, and the clinical and patient-reported outcomes are exploratory in that cohort. In the phase 3 cohort, we're actually in the process still of selecting the hierarchy of assessments. In other words, what exactly will be our primary endpoint, what will be our key secondary endpoints, what will be exploratory, and we're still working through that at the moment and crunching the numbers to tell us what has the most power. The good news is that we've seen directional improvements in multiple different endpoints, so we do actually have a lot of choice in what we can make as a primary endpoint. PROs are definitely tractable. FSHD isn't the only rare disease program that we have ongoing at the moment. We also have other programs in neuromuscular or muscular dystrophies, and we've actually disclosed outcomes from all three of our programs this year. And there's been a very positive reaction to that in the patient prescribing and also actually the investment community. So we have a program in myotonic dystrophy type 1. This is another typically adult onset muscular dystrophy that causes relentless wasting and loss of function over time, and ultimately for many, unfortunately, premature mortality. For that study, we disclosed 1-year data. So we had 1-year follow-up data that extended out through our open label extension study, where we again saw the very early improvements that we saw in multiple endpoints in the first study were maintained out over the 1-year period. And that data actually led to the design of our pivotal clinical study. So we actually have a phase 3 study already ongoing in that program that initiated in July of this year. So that's really exciting for those patients. And very recently, we had a readout on our Duchenne muscular dystrophy program. That's in Duchenne's, that's amenable to skipping exon 44. Duchenne muscular dystrophy is due to a massive deficiency or absence of the cytoskeletal protein dystrophin. There are drugs available to treat several of these different exon skipping amenable forms. There aren't any available at this point for exon 44. But normally what you've seen is dystrophin levels with therapy in the range of 1 to 5%. In our cohort, which is actually the lowest dose cohort at 5 mg per kg, we were able to show 32% of normal dystrophin. And the dystrophin that we make is near full length. So you'll have heard a lot about gene therapy for Duchenne muscular dystrophy recently. The dystrophin that's made there is a truncated form. It's a microdystrophin, whereas the dystrophin that we're making is near full length. And excitingly, in all of the patients, we saw a dramatic reduction in creatine kinase, this biomarker of muscle damage. In Duchenne muscular dystrophy, creatine kinase is typically in the thousands, sometimes even in the tens of thousands. And we reduce this down to near normal in every single patient, and that's been maintained over time. That's not been seen before, even with gene therapy, and certainly not with the exon skipping therapies. And then actually yesterday, so very recently, we disclosed that we're moving into a new therapeutic area with our antibody oligonucleotide conjugate approach into genetic cardiac diseases. So we call that precision cardiology. And yesterday we disclosed our first 2 targets for precision cardiology. So one of those is phospholamban, or PLN. This leads to a dilated cardiomyopathy, and the other target is PRKAG2. I won't give you the full name for that because it's a really long one, but that leads to a hypertrophic cardiomyopathy. But both of these are ultra rare genetic cardiac diseases that lead to very significant morbidity and sudden death in some patients. So that's super exciting. And so as a company, we're growing out, we're going into more diseases. We have a number of undisclosed exons skipping drugs, as well as exon 45. We're going to be moving into the clinic in the not too distant future. And we also are moving into other neuromuscular diseases as well, but we haven't yet disclosed those targets. So certainly lots going on at Avidity right now, but we have 3 programs in late stage development, all 3 of them targeting full approvals in the not too distant future. So that I think that's encouraging and hopeful for the patient communities.